Hi everyone, this is Heather Benson. I am switching to a new group, so for this week I'm on my own, but next week I should have a partner. So this is, I'm going to explain Lesson 8, Applying Genetics, and I'm going to share my screen here. Okay, so, oh, okay, so for problem number 1, it says, suppose that an individual is heterozygous for the following inversion. And if you look at up here, I show that. And then it says the normal chromosome and the inversion chromosome. And then it says, note that the alleles present on the chromosome are indicated using letters, and the centromere is indicated with a dot. And so I've indicated that here. And it says that they replicate before meiosis. Suppose that the two chromosomes replicate before meiosis. The two replicated chromosomes then attempt to synapse during prophase 1. And the question asks, what is the allele and centromere composition of the 4 products of meiosis if there is a crossover between S and T? So this concept's still a little bit complicated for me, but I'm hoping that I did this correctly. So what we have here is we have the normal chromosome, and we have the chromosome with inversion. This is paracentric because the inversion happens outside of the centromere. And it happens between these letters here, indicated in brackets. And so what we have, every time you have a paracentric pattern, you have one product that looks like one of these up here, one of the normal chromosomes. And then your next product is an acentric fragment, which means it has no centromeres. And that will look like this. XWVUT, SRQPOX, and then the product number 3 will be a dicentric chromosome, and that means that it has 2 centromeres. So we have this portion from the normal chromosome, this portion from the normal chromosome, and both of these portions come from the inversion chromosome, and they kind of come together to form that 3rd chromosome. And then the last chromosome is, it comes from one of these inversion chromosomes up here, so it's just the same thing. Okay, great. Moving on to number 2. Number 2 is up here. And non-disjunction in meiosis 2 produces 50% aneuploid gametes, whereas non-disjunction in mitosis can produce a mosaic phenotype. Meiosis in an individual who has suffered a paracentric inversion produces dicentric chromosome and a acentric chromosome. Part C, Turner syndrome is an example of a human disease caused by monosomy, while familial Down syndrome is an example of a disease caused by a Robertsonian translocation. For Part D, human liver cells can be 4N, 8N, or 16N. For Part E, a terminal deletion in chromosome 5 causes Cry-du-Chat syndrome, while an inversion on the X chromosome causes type A hemophilia. Part F, I wasn't entirely sure about, but I'm pretty sure that it's a small duplication is a change in chromosome structure that can be potentially beneficial to an organism or an entire species. And Part G, when a translocation cross is formed, the alternate segregation pattern can produce viable gametes and offspring. Okay, so number 3, it's a question concerning defects in chromosome number and structure. So how many total chromosomes would be found in the humans that have the following conditions? Turner syndrome would have 45 chromosomes. Jacob syndrome would have 47. Cry-du-Chat syndrome would have 46. Familial Down syndrome would have 47. and type A hemophilia would have 46. And remember that a normal human would have 46 chromosomes, and so some of these that have 46 aren't affected by adding or subtracting chromosomes. There's a different thing happening. Part B, which of the following terms is not associated with Charcot-Marie-Tooth disease type 1A? And that would be triploid, aneuploid, and trisomy because the chromosome number is not changed. Part C, which of the following terms are associated with Klinefelter syndrome? And I think that that would be aneuploid and trisomy and possibly non-disjunction. Great, so part D, which of the following terms are associated with type A hemophilia? That would be non-disjunction and Robertsonian translocation. Number 4, one pair of homologous chromosomes in a mouse has the following sequence of alleles. This here. And another pair of homologous chromosomes has the following sequence of alleles. And that's actually up here. Suppose a reciprocal translocation occurs between one chromosome in each pair, producing the following allele arrangement in 2 of the 4 chromosomes. That's right here. On one chromosome, and this arrangement here on the other, and there's 2 copies of that. Note that there's still one chromosome with this arrangement, and one chromosome with this arrangement. And we have the centromeres indicated here. Part A, diagram the synapses of these chromosomes in prophase 1. So what we have first is leptotina, and these are the top 2 chromosome pairs indicated up there. Next we have zygotina, which is where the synaptonomal complex forms, and that's forming right here. And so crossing over is going to occur. So we go over here to pachytina, and crossing over does occur, as we can show here on the tips. And then the complex here begins to dissociate, and we are left in the diakinesis stage with this chromosome and this chromosome, and this, and those are indicated up here. Okay. So, part B of number 4, identify the allele composition of the gametes produced by alternate segregation. Alternate segregation, if you look up here, let's see, we're going to call this chromosome 1 and this chromosome 2 because that's what happens in alternate segregation. It forms these chromosomes and another set, and then these chromosomes and another set. The crossing over occurs here. Then for part C, adjacent 1 segregation, what happens is we have part of chromosome 1 crossed over with part of chromosome, or with a normal chromosome 2. The second one is the same. On the third one, we have part of chromosome 2 crossed with chromosome 1 and chromosome 1, and the same as number 3. On adjacent 2 segregation, we have part of chromosome 1 crossed with chromosome 2, that's the ST, and then we have chromosome 1, and that's the same on part 2. And then the third set would be part of chromosome 2 crossed with chromosome 1, the K and the L, and part of chromosome, actually all of chromosome 2, and then number 4 is the same as number 3, so we have that set again. Okay, great. On number 5, it says, cats have a total of 38 chromosomes. How many total chromosomes would be found in the following types of cat? Part A asks for what would happen with a triploid cat. The equation for triploid is 3n. So up here I've calculated that cats have a total of 38 chromosomes. Divided by 2 equals 19. So 19 is the n value, and that's coming from one gamete. So 3n is 3 times 19, which is 57 chromosomes in a triploid cat. And then for part B, a cat that is monosomic for chromosome 12, it doesn't really matter what which chromosome number it is. What matters is that one of the chromosomes is monosomic, which means it has one less chromosome. So we have 2 times n minus 1. So 2 times 19 minus 1 is 37 chromosomes in a monosomic for chromosome 12 cat. Part C, a cat that has suffered a terminal deletion on chromosome 7. Terminal deletions do not change the amount of chromosomes that there are, only the composition. So what we have here is a diploid, because that's what they are normally. So we have 2n equals 2 times 19 equals 38 chromosomes in a cat that has suffered a terminal deletion on chromosome 7. Part D, a cat that has suffered a Robertsonian translocation between chromosome 17 and 18. And so what that means is this is what happens with familial Down syndrome. So we have an additional chromosome. So it's 2n plus 1 equals 2 times 19 plus 1 equals 39 chromosomes. And finally, part E, they want to know how many chromosomes there would be in a cat that has suffered a reciprocal translocation between chromosomes 6 and 9. And as with part C, a reciprocal translocation does not change the total number of chromosomes, only the composition of the chromosomes. So it would be diploid, 2n equals 2 times 19 equals 38 chromosomes. I hope that made sense, and thank you for watching.